In this podcast episode, Joe Rogan welcomes Beth Shapiro, a pioneering scientist in the field of ancient DNA, also known as paleogenomics. Beth explains that her work involves extracting DNA from ancient remains, often digging up bones and other materials to uncover genetic information that can rewrite our understanding of history and evolution. She describes her role as a modern-day explorer, uncovering new insights that challenge established scientific narratives. Beth shares her journey into science, which began unexpectedly after an initial interest in broadcast journalism. A transformative field geology and archaeology course sparked her fascination with human history and the natural world, leading her to pursue a scientific career. She recounts meeting Alan Cooper, a pioneer in ancient DNA research, who invited her to join his lab and travel to Siberia, a trip that solidified her passion for the field. This blend of adventure, science, and storytelling defines her unique approach to research and communication. Beth discusses the scarcity mindset prevalent in academia, where competition for funding and recognition often fosters negativity and gatekeeping. She explains how this environment can stifle innovation, as researchers may resist new ideas or collaborations that threaten their own status or resources. This culture of exclusivity sometimes leads to dismissive attitudes toward groundbreaking work, especially in controversial fields like ancient DNA and de-extinction. Podcasts and other modern communication platforms have disrupted traditional academic gatekeeping by allowing scientists like Beth to reach broader audiences directly. However, this visibility also invites pushback from colleagues who may feel threatened or skeptical. Beth reflects on the tension between maintaining scientific rigor and engaging with the public in accessible ways, emphasizing the importance of open dialogue despite criticism. Beth shares vivid stories from her fieldwork in Siberia and northern Alaska, highlighting the extreme conditions and unique challenges of working in these remote environments. She describes the relentless mosquitoes that plague researchers during the short summer season, recounting humorous and harrowing encounters with swarms so dense they can kill dozens with a single clap. These insects are adapted to the Arctic climate, capable of reproducing without blood meals, which adds to their ferocity. Her experiences also include logistical difficulties, such as unreliable helicopters, unpredictable weather, and encounters with indigenous communities like the Dolgan people. Beth recounts how these subsistence hunters live in harmony with the harsh tundra environment, herding reindeer and maintaining traditional lifestyles despite modern pressures. These field stories underscore the blend of scientific inquiry and cultural respect that shapes her work. Throughout the conversation, Beth emphasizes the resilience and wisdom of indigenous peoples who continue to live in traditional ways across the Arctic and other remote regions. She highlights their deep connection to the land, their sustainable herding of reindeer, and their efforts to revive native languages suppressed during the communist era. These communities embody a way of life that contrasts sharply with modern urban existence, yet they often exhibit a profound sense of fulfillment and happiness. Beth also touches on the complex relationship between indigenous groups and outsiders, including the risks faced by uncontacted tribes in places like the Amazon. She acknowledges the ethical dilemmas involved in scientific exploration and conservation, stressing the need to respect these cultures and their autonomy while learning from their knowledge of the natural world. Beth provides an accessible explanation of the technical challenges in ancient DNA research, such as the degradation of genetic material over time and contamination risks from modern DNA. She describes the stringent laboratory protocols required to prevent contamination, including working in clean rooms with protective suits and specialized airflow systems. The advent of PCR, polymerase chain reaction, revolutionized the field by enabling the amplification of tiny, fragmented DNA sequences extracted from ancient specimens. She recounts early controversies in the field, including false claims of dinosaur DNA that were later debunked as contamination from modern sources like chicken DNA. The successful sequencing of Neanderthal and Denisovan genomes marked major milestones, revealing unexpected interbreeding with modern humans and reshaping our understanding of human evolution. Beth highlights the ongoing potential of ancient DNA to uncover hidden chapters of natural history. A significant portion of the discussion centers on Colossal's groundbreaking work in de-extinction, particularly their efforts to bring back the direwolf, an extinct species that lived alongside early humans. Beth explains that the direwolves currently being developed are genetically engineered gray wolves with specific direwolf traits reintroduced through genome editing. This approach allows them to recreate animals that closely resemble the extinct species without cloning from intact ancient DNA, which is unavailable. Beth describes her emotional experience witnessing the birth of Kessie, the first direwolf pup, and the awe inspired by seeing these animals alive after thousands of years. She acknowledges the ethical and ecological questions surrounding de-extinction, including concerns about ecosystem impacts and the responsibilities humans bear as stewards of revived species. The Direwolf project serves as a test case for the broader possibilities and challenges of species restoration. Beyond de-extinction, Beth discusses how the technologies developed at Colossal and in ancient DNA research can aid conservation efforts for endangered species. She highlights the example of the Florida panther, where introducing genetically related individuals from Texas helped alleviate inbreeding problems and revive the population. Similarly, Colossal is working on red wolf conservation, using genetic tools to increase diversity and strengthen the species' survival prospects. Beth stresses that these interventions are forms of genetic rescue, aimed at enhancing the resilience of threatened populations facing habitat loss, disease, and climate change. She envisions a future where gene editing and genome sequencing become integral parts of wildlife management, helping species adapt to rapidly changing environments and preventing extinctions. The conversation delves into the scientific complexities of defining species, especially in light of widespread hybridization among closely related animals. Beth explains that traditional species concepts, such as the biological species concept based on reproductive isolation, often fail to capture the fluidity of genetic exchange observed in nature. Examples include the interbreeding of polar bears and brown bears, as well as domestic dogs and wolves. She points out that many species boundaries are porous, with gene flow occurring between populations previously thought to be distinct. This challenges rigid taxonomic classifications and calls for a more nuanced understanding of species as dynamic entities shaped by evolutionary history. Beth uses these examples to counter criticisms that de-extinction projects create fake animals, emphasizing that natural hybridization blurs such distinctions. Beth shares fascinating insights from ancient DNA studies on human evolution, including the discovery that modern humans carry Neanderthal DNA, which contributes to various traits and disease susceptibilities. She notes that by aggregating Neanderthal genetic fragments from diverse human populations, scientists can reconstruct a substantial portion of the Neanderthal genome, raising philosophical questions about extinction and identity. Recent findings of previously unknown human lineages in South America and other regions further complicate the picture of human migration and diversity. Beth highlights how ancient DNA continues to rewrite human history, revealing complex patterns of interbreeding, migration, and adaptation that challenge simplistic narratives taught in schools. Throughout the episode, Beth addresses the ethical dimensions of gene editing and de-extinction. She acknowledges public concerns about playing God and the potential risks of manipulating genomes without fully understanding long-term consequences. However, she argues that humans have always shaped nature, from domestication to habitat alteration, and that responsible use of new technologies can help repair past damage. Beth emphasizes the importance of transparency, regulation, and interdisciplinary collaboration in advancing gene editing applications. She cites recent medical breakthroughs, such as gene therapy for rare diseases, as examples of the positive potential of these tools. Looking ahead, she envisions a future where gene editing supports biodiversity, human health, and ecological balance, provided society approaches it thoughtfully. In closing, Beth reflects on the broader relationship between science, society, and innovation. She critiques the current academic and political climate where funding scarcity, ideological conflicts, and misinformation hinder progress. Beth advocates for open-mindedness, critical thinking, and constructive dialogue to overcome these barriers. She also highlights the transformative power of technology and the need to embrace change while respecting ethical boundaries.